Welcome to video 14 of Genetics the Code Broken. This is the final video in the series. In this video, we're going to be looking at how genes have evolved over time and how this provides evidence for evolution. So briefly at the end of the last video, we discussed how many of the sequences and many of the structures and many of the processes that cells undertake are similar, no matter what cell we are looking at. Because there are lots of genes in our body that contain many of the same sequences, we can say that they have only slightly varied over time. If we compare the gene sequences of humans to many other organisms that are on our Earth, we can see that they have only changed slightly over time, and this has happened over a period of millions of years and over the time which we've seen a great evolution in the amount and the variety of organisms on Earth. So sequences that do not change or change only very slowly over time are said to be conserved, and these are the genes that we use to measure phylogenetic relationships, which is the degree of relatedness of an organism. The nucleotide changes that are involved are usually neutral mutations, which means that although the sequences have changed slightly, the changes that have occurred do not affect the final functioning of the amino acid produced. If we go back to looking at Hox genes, we can see that the homeobox sequence is conserved. So it is thought that they are homologous genes that share a common ancestral gene. And it seems highly likely, we can use this diagram on this slide to show that animal species inherited all of their Hox clusters from a common ancestor. So we can see from the picture here that all Hox genes are homologous, and the sequences which we are shown in blue are very close homologs. So ribosomal RNA is coded for by genes, and these have only changed slowly over time, where in contrast, mitochondrial DNA changes rapidly and therefore can only be used to compare organisms that have diverged relatively recently. In terms of molecular clocks, we're looking at the rate of change or the mutation rate of a gene is known, then we can compare the nucleotide sequences in the same gene in different species. By using these molecular clock calculations, we can use these to estimate how long ago they shared a common ancestor. Because this technique uses DNA analysis, it is only currently used to compare living species, and the data that has been calculated so far does correlate with fossil evidence that we do have to those species that no longer exist and that are extinct. These molecular clock calculations have also been used to compare mitochondrial DNA, suggesting that the modern humans may have originated in Africa about 100,000 to 200,000 years ago, which is a lot more information that is previously known. In terms of evolutionary relationships, we can study the mutations of the homeotic genes, as we've mentioned previously, and we can show that a small mutation can in fact result in a dramatic and sudden change in an organism, and that is if the developmental cascade is altered. Gene changes or evolution can result in the evolution of new body structures, and it can happen rapidly, suggesting this as a mechanism for the evolution of the diversity of living organisms. And if we look at this gene change in closer detail, we can see that it adds support to the punctuated equilibrium theory of evolution. Let's conclude video 14. Let's make sure that we finish all these worksheets and get this HSC done. Congratulations on finishing the HSC biology course.